So I'm Parthiba Basu from Center for Pollination Studies and Department of Zoology in University of Calcutta, which is in India. We have started this center to study the status and the roles of pollinators in Indian agricultural system, basically, which is part of a Darwin Initiative project. And as part of that, we are looking into the status of various pollinators, which are very little known, really, what they do in the Indian farming landscape, if Indian farming system is pollinator limited, etc. These kinds of questions. Although we do very strongly believe, based on our analysis of secondary data, that India definitely has a pollinator limitation crisis, there is really no first-hand data to prove that, and we are just trying to do that in number of Eastern Indian agricultural landscapes. We have also been trying to reach out to the farmers and make them aware about the roles of pollinators and why do we need to save pollinators, and trying to develop conservation programs involving the farming community in all these three states. Alongside, we are also interested in looking at the impact of various drivers that's affecting the pollinators. And as is quite well known, that pesticide being one of the major drivers that people talk about, we are definitely interested in looking at what kind of impact does pesticide or do various kinds of pesticides have on the important pollinators. So honeybees there are many species of wild bees, and they are very important for pollination, but in terms of effective pollination, honeybees are really important ones. And in India we have two major species. One has two subspecies, so two major species of honeybees, one that are domesticated and one of them is domesticated, one is not, which are hive making bees. So one is Apis dorsata, which is a rock bee, which is quite aggressive and cannot be domesticated, but they play a major role in pollination. The other one is Apis cerana, and Apis cerana with the two subspecies of that, indica and cerana. And they're domesticated as well. There are other couple of species who also are good pollinators, but not as much in terms of importance as these two species. One is Apis florea, another is trigona. So there's two minor ones, but these two are the major ones. Wanted to see what impact pesticides have on these species. Although pesticides, in intensive agricultural landscape, and India has had its run of green revolution, and for the last 45 years or so, close to about 50 years now, there is high-scale agricultural intensification across the country, and in Eastern India is another hotbed, although it had green revolution a little later, but it's now quite intensive agriculture in large vast tracts, and that of course is associated with huge pesticide application, disappearance of semi-natural vegetation from the landscape, and that's been aggravated after the new economic liberalization post-90s, with a lot of thrust on urbanization and stuff like that, new industrialization. So we are losing, at a very fast rate, we are losing a lot of our semi-natural vegetation from our landscapes. We figured that it should have impact, but what's the extent of this impact needs to be understood, because you still do find beehives in the landscape. So probably they're coping somehow. Of course, one needs to understand when would be that last straw that would break the back. But so we need to understand the extent of the damage that has taken place. So we had three major hypotheses. I mean, so one was pesticides definitely can impact their behavior, right? Potentially can impact their behavior, and behavior would mean their ability to homing behavior or foraging behavior, etc., etc. And for foraging or homing, either way, olfaction and vision, they are two very important factors that would regulate that. And also at physiological level, I mean, there would be signs of stress. Pesticides worldwide is known to cause oxidative stress. And oxidative stress is like, well, things get rusty, right? Your cells get rusty. So you need the kind of oxygen that you would require. To do a simple task, you would require more oxygen, right? So that's oxidative stress. Honeybees, when they get oxidative stress, so that would definitely affect their orientation, their homing behavior, their foraging behavior, and of course, this pesticides definitely has an effect on their larvae, their brood, and in fact, that's another round of investigation that we are doing right now, looking at what's happening to their broods and how their broods are affected, because some of the deformations in their antennal sensillae, that looks like quite obviously their developmental abnormalities. So we started off by choosing an agricultural intensification gradient. We sampled honeybees from agricultural landscape that's been into intensive agriculture for some time, quite some time, and then we chose agricultural areas which are very close to forest, very little pesticide application, etc., and also sampled bees from there. And we compared the levels of oxidative stress in these two kinds of bee populations, sampled from the field itself. There are a number of markers, number of enzymes that people use to understand if there is any stress or not. So we use three of them. One is one directly shows if there is oxidative stress. So this particular enzyme, it shoots up. Okay, so it's a stress marker really. It shows that there is stress. Okay, and name of this enzyme is xanthine oxidase, if that's of any interest to anyone. The other two, they are basically antioxidative enzymes. So if there is stress in the body will try to cope. And so if there is stress, so these two other antioxidative enzymes are also supposed to shoot up because your body is trying to cope. Okay? So we also chose two of those antioxidant enzymes. So one is called superoxide dismutase and one is called catalase. So these two antioxidant enzymes. And we measured these three and compared two populations. What we found was in the pesticide zone, the samples from pesticide zones, it's significantly high oxidative stress in terms of all these three markers. And so the honeybees in the pesticide zones, they have heavy oxidative stress. There's a very simple behavioral tool that people use to see behavioral change, mostly for olfaction, mostly for smelling. Okay, so generally if they're excited, if the insects or the bees are excited, they would take their little tongue out as response to that lure or whatever, that attractant. Okay, the proboscis would come out. So if their smelling power is sort of subdued, then they won't be taking out their tongue, would they? Right? So we did this experiment, and train them to take out their tongue, etc., before and after exposing them to pesticides in controlled experiment to start with. And with pesticide exposure, no, they were not quite taking out their tongue, because so they were not excited. They could not catch that attractant odor. Okay, the sample again, bees from the pesticide zones, they also had the same problem. They could not quite recognize the odor. They could not take out their tongue. They did not, right? So, one first sign that their smelling power is somewhere going wrong, has gone wrong somewhere, right? Their smelling organ is in their little antenna, okay? And this antenna have many small sort of structures which are called sensillae. And the role of sensillae is to sense, okay, micro structures. And there are various kinds of sensillae in the bee antenna, and of them there are about 10 such sensillae types that are responsible for smelling. Okay. So we again checked the antennae of honeybees from the pesticide intensive zones and places where there is no pesticide application from the agricultural landscape. And what we found we were quite appalled to see that the sensillae of the bees sampled from the pesticide intensive zones, they are quite heavily deformed, really. There is one particular one which I must mention. There is a particular one which it looks like a disc, and this disc shaped sensillae in the pesticide zone bees had real crack marks, okay, like a chaffed dry soil. Okay, it's got crack marks, little sort of bristles, which would in a healthy bee would be quite long and curvy. It's got sort of diminished and deformed. So their behavior is changed, and their sensillae, their sensory structure is also deformed. Now, when they sense something, the sensillae, when they sense something, that got to be processed somewhere in the brain. And in honeybee brain, there are two. So the two regions where the antenna connect, they call the antennal node in the brain, and there's a mushroom body, a little tiny structure in the brain, where all these informations are processed, and then they decide that this is that smell and that is that smell, right? And calcium is a very important commodity that plays a very important role in this memory retention, this smelling memory retention. So we wanted to check the health of this calcium process in their brain. There are many ways very sophisticated ways of measuring such calcium channel, etc., with microsensory probes put in their brain, etc. We didn't take that path. That's the slightly more sophisticated and more expensive path for exploration. But thankfully we have now beautiful microscopes that can image the process, okay, various molecular chemical process. And so we did something very bad. We dissected out the brains of live bees and injected some dyes which would fix this calcium in their brain and let them live for some time. And we had to sacrifice some of them for this to come out, to get the truth out. And then we took a section of them and and we looked under the microscope with all these fluorescent dyes. So if there is high calcium, free to bound calcium is with this dye. So free to bound calcium, this would give a particular very light green sort of a fluorescence under the microscope. If there is good amount of calcium doing round in the brain, that means their olfaction is all right. And if the smelling power is not all right, then there won't be much free calcium doing round in the brain anyway. Okay? In the high pesticide zones, you didn't have those nice green fluorescence that you were looking for, because their calcium channel is blocked. So their sensilla deformed, and whatever little information they were getting, they're not able to process because the calcium channel is blocked as well, right? And then, of course, there was a little bit left, and one of our sort of good reviewer friend pointed it out, said, yeah, okay, so try that little bit extra. We need that information to seal this fact. So calcium is, so the certain genes which are responsible for expressing this, calcium production, etc., is called calpain. So see if there is this calpain expression is upregulated or downregulated, and we again check that out. And obviously in pesticide zone, the calpain was quite downregulated, and they're quite normal upregulation, normal regulation in the pesticide-free zone. So summarizing all this up, pesticides are creating havoc for these honeybees, both species, Apis dorsata and Apis cerana, in the pesticide-intensive agricultural landscapes in India, where we have sampled, where we have studied. And this, we think, I mean, there is a threshold somewhere that's still somehow able to cope, but it's not going to be a long-lasting story. And they are disappearing. So we also had held some sort of discussions with the farming community, and the problem is, in many countries, most countries, you do not have a good baseline data to compare the present status of population. So, but we did some extensive sort of discussions with the farming communities, and interestingly, they all say that they can identify a number of bee species when we showed them the picture, etc. And also they know very well these two honeybee species. So their population has significantly dropped over the last at least couple of decades. Okay, so definitely their populations are going down, and pesticide has really played a major role in that. And so hopefully this would ring a bell somewhere at the level of the policy making, that pesticide application is regulated severely to save the pollinators.